Hi, I'm Bob Vonderheit. I'm the director of the Abramson Cancer Center here at Penn, and welcome to the annual Vassar Center BRCA Breakthroughs and Discoveries panel. I hope you enjoyed the previous session where that was for the lay audience, and we're really going to dig into the science. This has become a really special part of the symposium where we get to talk shop, and particularly in a more general way. And the panel will be chaired by Susan Domchek, the executive director of the Vassar Center, and she'll introduce her guests in a minute. I just want to take one moment from the point of view of the Abramson Cancer Center and Penn Medicine to thank and be sure to express our forever gratitude to Mindy and John. Mindy is not here in person, but listening, and we'll hear from her shortly, I think, if we got the technology worked out. Mindy and John, thank you for your vision. It's 10 years, and it's pretty amazing, right? And we met Sherry and Len earlier in the program, and we welcome them here as well, and thank you to both of you for your incredible contributions and support in so many ways, not just to the Vassar Center, but as you know, to other aspects of the Abramson Cancer Center. So it is amazing to think about the 10 years and all the science that's happened. We have PARP inhibitors. I think what's amazing is to watch how this has become a conversation in the community and the population at large. I think the Vassar Center has had a lot to do with that. We heard about Chris Everett, who heard about her sister and realized she needed to get tested, so now she was in the news today about her own story, and we see that again and again. An incredible innovation getting the word out, not just to individuals who might be at risk, but also to doctors, and also finding out ways to deliver the genetic information directly to the electronic health record. There's been a lot of quiet innovation as well that's making a huge difference. And so we're really proud of that work from all the way from the basic to the clinic to the population at large. I think it's making a big difference. I want to now ask Mindy to come forward, Sherry, and also Mindy to tune in, I guess, and to take the stage and to introduce this year's Pearl and Philip Vassar Innovation Research Award, which is given annually to a young investigator on the cusp of greatness. Sherry.
Mindy.
Hi, everyone. Sherry and my father, Phil, or Philadelphia Phil, as some of you may know him, turned 104 in March. He has lived a life filled with the highest of highs and the lowest of lows. Raised in an orphanage, served in World War II, lost a daughter to BRCA cancer, and then a wife to lung cancer. Our late mother, Pearl, did not have an easy path either. A daughter of two deaf parents who are recent immigrants struggling to make ends meet in Philadelphia, she was forced to grow up way too soon. But despite their modest backgrounds, our parents instilled in us the value of education, gratitude, and tzedakah, which in Hebrew means giving back. The Pearl and Philip Vassar Innovation Research Award is rooted in these values and endowed thanks to their incredible support and dedication to the Vassar Center. It honors the memory of their daughter and our sister, Faith Vassar, while also demonstrating their hope and commitment to ensuring other families would not face the unimaginable loss our family will forever mourn. This award provides the recipient with a one-year, unrestricted research grant, providing young faculty and investigators with the resources to advance breakthrough science, publish findings, and pursue larger grants. By investing in this type of innovative research, Vassar is planting the seeds for the next generation of therapies and prevention. We are hoping these bloom into better treatment options and ultimately an end to BRCA cancers. Thank you. And now, my sister Sherry, who I'm sad not to be there next to in person.
Well, we definitely miss you, Mindy, but we are so thrilled to announce Dr. Payal Shah is the recipient of the 2022 Pearl and Philip Vassar Innovation Research Award. Dr. Shah is a brilliant medical oncologist and clinical investigator at the Abramson Cancer Center. Dr. Shah attended Penn Medical School and completed her residency here too. She is a homegrown talent. She has also been an integral part of the Vassar Center team for over 5 years. Dr. Shah's work focuses on developing new treatments for breast and ovarian cancer patients, including those with inherited cancer. This work includes the HOPE trial, which examines the safety and efficacy of a new combination therapy for women with BRCA-related metastatic breast cancer, as well as a clinical trial administering CAR T-cell therapy in patients with ovarian cancer. Dr. Shah has presented her work at many national meetings and has published in several prestigious scientific journals. She's also participated in several information centers for Vassar that help raise awareness of the impact of BRCA mutations. Along with Sadaka, our parents also emphasize the importance of being kind, hardworking, and honest in your dealings with people. Dr. Shah is one of the warmest, most personable, and empathetic doctors you will meet. Her patients rave about her ability to explain things in layman's terms in a gentle, comforting, and respectful manner. These are all the traits that represent everything our parents stand for. It also doesn't hurt that Dr. Shah is a brilliant female scientist forging transformational discoveries that will lay the foundation for tomorrow's breakthroughs. We are so pleased to support her work and very excited to invite Dr. Shah to join us to receive her award. Congratulations.
Thank you so much. Thank you, Sherry, Mindy, and thank you so much to Dr. Shah, who's I'm so lucky to work with. So, congratulations. So this discussion, again, we're going to be turning from the deep science to more of a lay public-facing discussion. We obviously can't answer specific medical questions, but we're going to try to take as many questions as we can. These can be put through by Slido, or you see that QR code? I think we've all figured this out from menus over the pandemic. So if you take a picture of it, that'll take you to where you can put in a question. And so we'll be doing all of that as we go. But first, we're just going to start. I'm going to introduce our panelists. We're going to have a discussion, and then we're going to launch into your questions. Of note, a lot of people put their questions in advance, so I've tried to kind of incorporate those questions into our initial discussion. All right, so let's get started. So first of all, I'd like to introduce our panel. To the immediately left of me, for those of you who were at our last session, is Andrew Nussenzweig, who is of the National Cancer Institute. He serves as the branch chief of the Laboratory of Genome Integrity in the NCI Center for Cancer Research, and he's this year's Vassar Global Prize recipient, gave today's keynote. Right next to him is Dr. Kara Maxwell, who's an assistant professor in medical oncology here at Penn Medicine. Dr. Maxwell, I've been lucky to work with her since she was a fellow. Now she has her own independent laboratory and is focusing particularly on BRCA-related prostate and other cancers. And Dr. Andrew Tutt is the director of Breast Cancer Now Research Center at the Institute for Cancer Research in London. Dr. Tutt is the consummate sort of triple threat. He is a clinician, a basic laboratory researcher, and a clinical researcher as well. He's focused on triple-negative breast cancer as well as BRCA-related breast cancer, and we've known each other for probably longer than we should let on. I'm also pleased to announce that as of today, we received a significant grant funding that has been established for the Men and BRCA program, established through a generous philanthropic gift. This is going to allow us to invest in our research and education efforts specifically on men's issues. Kara will be leading that program for Vassar, and I know that she has ambitious goals and research plans in this expanded area of focus. I think all of us in this room know that if you lined up all the mutation carriers in the world, 50% would be men, and yet it is not true that 50% of mutation carriers that currently know it are men. So we still have a lot of work to do in that space. So right now, I'm going to get us started by just asking each of you, just for 2 or 3 minutes, tell us about your work and what you do. So Andre, you first.
So I study a type of DNA damage called a double strand. That's a double strand break where both strands are interrupted very close by to each other. That generates this DNA breakage, and this is relevant for BRCA deficiency because they're unable to repair those double-stranded breaks. And as a result, these breaks accumulate, you get mutation, and this predisposes to cancer. It also leads to a vulnerability in that these deficient cells now are sensitive to DNA damaging agents that produce double-strand breaks. So loss of repair leads to cancer, but also leads to a vulnerability.
Great. And Kara?
Thank you, Susan. So in relation to my work in BRCA, I'm just thrilled about the new Men's Center, so thanks for bringing that up today. And that focuses on some of the work that I do in my lab, working on BRCA-related prostate cancer. So we're very interested in sort of mechanisms of early development of cancers in men with BRCA mutations. And through this work, I'm leveraging a really great network of collaborators within the Veterans Affairs. We have a large network of folks, and we're actively biobanking for this because one of the questions is for our men, mostly our men who served, for example, in Vietnam, those exposures that they got, well, how does that interact with, say, a DNA repair mutation that could lead to the really significant amount of cancers, including prostate, that we see in our veteran population. I also study another rare syndrome called Li-Fraumeni syndrome. It's a multi-cancer syndrome, and in that, we're interested in early breast development, but that's a little bit less relevant for today. So
you never know what questions we'll get.
You
so
never
maybe
know.
We'll get back to you. And Andrew?
So as a young trainee oncologist in the mid-1990s, I got to do a PhD in a basic biology lab with my mentor, Alan Ashworth, and to get to work on what the BRCA2 gene function was. So what I've loved to do since is to try to connect physicians, oncologists with that basic fundamental science mission, and have been privileged to be involved in development of therapies like PARP inhibition. So what do I do now? I continue to get very excited by trying to translate and connect between fundamental biology and clinical trials development. And I run a joint lab with my colleague Chris Lord in trying to understand where we can use approaches like PARP inhibition and the new therapies that might combine with it best, and where we fail, why we fail, and to work with fundamental scientists like Andre and many who are at this meeting to try and bring the latest fundamental biology understanding to the clinical trial. Reverse translation meets forward translation, as we call it, and to do that in a big global translational environment. That's how I work with Susan and many others, so that's me.
And so it does give you a sense of sort of the spectrum of research that we need to get done. I, for instance, don't pipette at all. I don't have what we call a wet lab. I have a lot of people that I work with in research, but we use computers and we collect samples and we do clinical trials, and it takes all of us to move the field forward, and it works best when we collaborate together. But I'm going to take a step back. So, Andre, one of the biggest things that people often say is, I have BRCA. What they mean is that I have a BRCA mutation. So tell us, every cell has BRCA1 and BRCA2. So just tell us more about that.
What is the question, though?
You told us a little bit about what
Yeah.
they do in the
Yeah.
cell, but
Yeah.
you've talked a little bit more about what mutations might do. But tell us a little bit more about normal function of BRCA
okay.
proteins.
Yeah. So normally, BRCA proteins function in a complicated pathway called homologous recombination, where the break uses a in one chromosome uses the intact allele to copy and repair that break. And that's really a fundamental part of any cell that's in cell cycle can have random DNA breaks, and it utilizes this pathway to repair those breaks.
Yeah, I mean, we can't emphasize it enough. You know, we all have BRCA1 and BRCA2, and we're happy to have them. You know, if we didn't have them, that would be bad. And so oftentimes we just use slightly the different wording. It's when someone has an alteration or a mutation or a pathogenic variant. Unfortunately, we use all these terms these days, but they all mean the same thing, which is a BRCA gene that expresses a protein that doesn't function. And that's when, if you will, we get into trouble. So these genes were identified and cloned before we knew what the basic biology was. And I was not involved in it, when it was figured out that what they were, we realized that they were, loss of function, that the mutations made basically the protein not expressed. So when we think about targeting cancer, we often think about targeting things that are turned on, HER2/neu, estrogen receptor. They're easier to kind of conceptually think about. But Andrew, you were involved in figuring out, along with others, about how to target things that were turned off. So tell us how you did that, as long as others, because I know you're always humble.
As the little guy in the lab who was the clinician.
Who did it all.
Yeah,
yes.
I got to watch a lot of other people do it, but and be involved in application, I guess. So I think you're right. I think it is a concept that people, at least at the time, it was relatively alien, that you were usually thinking about targeting a strength, not targeting a weakness. And so I think it was, considering this a kind of an Achilles heel, it was a strength for the cancer, that it had got this way of getting all of the mutations it needed to be a fully fledged cancer more quickly, and that meant higher risk and earlier. But the Achilles heel in Achilles was it left behind in the cancer this weakness that came with it. And how could we find a way of targeting that residual weakness that the tumor still had? I think what the work of Andre, who beautifully described it, and he described the work of many others before, has led us to understand, how do we then, having learnt to target it with PARP inhibition, there are a number of ways in which Achilles puts something back over his heel and protects it, re-enables. And I think that the power of this kind of collaboration is to really understand what those things are, and do they create another vulnerability? Does Achilles, in doing that, does he gain another weakness in his armor somewhere? Or how can we target it? And I think that's what the field is now very exciting. It's very exciting that this is happening. It was only when we, the royal we, when the field began to be able to develop these approaches, develop these drugs, and for some people be successful, would we expose resistance, expose where it didn't work, and that then creates the new challenge. And the collaboration with science helps us understand where what we know can happen in the lab does happen in the clinic, and that's what we should focus on trying to address for people who have these genetic forms of cancer.
Yeah, and to emphasize, the specific biologic things that are seen in modeling in the lab, do they happen in the clinic? And then what do we see in the clinic, and how do we model that in the lab? And it has to go both ways, bench to bedside and back, as we say. We're going to talk more about PARP inhibitors, because the leap from understanding the basic biologic principles to a therapeutic target still actually makes my head spin that this has happened. And so it's very exciting. But first, we know a lot about BRCA1 and 2 mutations, or again, pathogenic variants, and the associated risk of breast cancer and ovarian cancer. But Kara, there are other cancers. So tell us more about that.
Yeah, and it's kind of nice. Was just another JCO paper by Antonin and his colleagues that just came out. That's we think we know the answer, but then we keep learning new things. And so this paper really confirmed what we know, that individuals have elevated risks of breast and ovarian cancer. But what was really interesting in this recent paper that came out was that it was really BRCA2 carriers that had an increased risk of prostate cancer. And I think that that's going to, kind of showing up over and over again. Do BRCA1 carriers have the same risk? And, it might not be the case. And some of the work we've done in my lab would suggest that as well. But what I think this paper then did too was think about what are some of the other cancers that we might be at risk for. And these are complicated. These are large epidemiological studies, and things often get, I think, sensationalized in the news, and we have to kind of dig deep to see if we're going to add a new cancer to the bucket of BRCA-related cancers, we better be really sure. And so what this recent paper suggested is perhaps there's an increased risk of stomach cancer. And I think that that's one of the things that we're really interested in, and hopefully the Men's Center can help work on that with my colleague Bryson Katona. He's very interested in this as well, because we know that there's a lot of sort of misinformation in the health record. And one clue in that paper was that women seem to be at more risk than men for stomach cancer. Well, that doesn't seem to make sense, right? Because we know men in general are in more increased risk of stomach cancer. So there may be some misreporting in the health record that's going on there. So I think that's going to be super important to tease out. And we've done some work also in my lab looking at other non-canonical BRCA tumors to see, are they really driven? Just because you have a mutation in a gene and you get cancer doesn't necessarily mean that those two are related, that the one caused the other. I think that it's crazy to think, but it's still an open question.
And I mean, just to be clear, that what we think about that association that was that Kara just mentioned is that these were all self-report on family history, so they were probably all ovarian cancers. That's why it was that's why it may have looked like it was more in women. So just I saw Sherry's face even behind the mask, and I just want to say we're going to investigate that more, but it's one of these things that we have to always take our clues and follow the data where they lie. But pancreatic cancer is the other cancer that we'll get back to as well. But so let's talk about the good news first about PARP inhibitors, and then get a little into the resistance issues. So the great news about PARP inhibitors is that these are FDA-approved medications for breast cancer, ovarian cancer, prostate cancer, pancreatic cancer, and that's crazy because the first approval happened in 2014. And so it's just been such an exciting time, but it became even more exciting over the last year because of the Olympia trial. And so, Andrew, tell us about the Olympia trial.
So it's been the most incredible privilege to lead this trial, which was actually, when we look back and think about it, Olympia is a trial that said very early we had a concept that we thought we'd proven in the lab and in animal experiments. The group of scientists had worked on this with clinicians. Could we go very rapidly to ask, in women who had genetic forms of breast cancer, BRCA1 and 2, but had a curative setting, so they were having their surgery, their chemotherapy, but were still at risk of recurrence, could we apply a PARP inhibitor then and improve survival? We hadn't even got a license for PARP inhibition in advanced ovarian, any of the indications you just mentioned, advanced ovarian cancer, prostate, breast, pancreas at that point. And this trial was put together across the groups of the NCI, the Breast International Group, genetic oncologists. I mean, Susan was very, very much involved and still is. And that trial has just reported its results last year and has just re-reported survival results showing substantial improvements, reductions in about a third of the risk of death caused by having a year of tablet-based treatment with olaparib in this situation. And that's led to an immediate FDA license in the first curative intent treatment context, all based on understanding that just because it's a rare form of breast cancer, it's still an important group of patients, women, to study internationally. 700 hospitals collaborating, each actually only recruiting a very small number of people, but saying this is important, this is what the science says, let's move fast and do this, and this is the result. So I could not be more pleased on behalf of the science, the collaborative, and most importantly, the families and patients.
And just to emphasize that, over 1,800 women and men across the world enrolled onto this study, knowing that they were 50% chance they'd get drug or a placebo, but knowing that this was crucially important for us to get the answer. It was possible that it didn't help and it hurt them. It was possible that it helps and didn't hurt, and we just didn't know the answer. And so I always am astonished at really the decisions that our patients make. And so the other thing I'm going to say is that Andrew gave the initial plenary at ASCO last year, and it was just such a shame because it was virtual. And this is the type of plenary where it absolutely would have gotten a standing ovation, because this is a really big advance in the field. And someone actually submitted the question, can you discuss the changes that have been made to treatment protocols based on Olympia? And the answer is, if you meet the criteria in the Olympia trial, which is a tumor in triple-negative breast cancer associated with the BRCA1 or 2 mutation, if it's high risk enough, or ER-positive disease and high risk enough, after you get all your other therapy, you should get a year of olaparib. So you should ask your doctors, is this the right medication for them? But really, it actually de facto has changed the standard of care for how we treat people in the United States. I don't know if you can give it in Europe yet.
We can on a expanded access program in a number of countries made available by the pharma involved. So yes, our patients in our NHS in the UK, in my hospital in London, are able to receive this based on that guidance. If I could just say one other thing that I think has where this collaborative effort has changed guidance is this is now something that treating physicians know they need to know about to advise to inform the conversation with their patients, not just a discussion that might become relevant at some point to think about familial risk or second breast cancer risk. This is now information just like they need to know what the HER2 status is of a cancer or the hormone receptor status. This is important to know, and I think that is driving the whole thinking about genetic testing, having conversations with people about their family history, and immediately after the plenary last year, before the FDA license, which was only in March this year, the guidance on genetic testing had changed in the US and internationally based on the fact there was a reason to do it because of this therapy trial. So it has.
Yeah, the one thing that we were discussing yesterday is, whether this treatment can prevent other cancers like ovarian. Will it turn out that by treating with a PARP inhibitor, what is the impact on ovarian cancer in these prone in these carriers? And when will you know about that?
So I mean that is an incredibly important question, and one that we knew that a trial like Olympia would be able to address as what we call a secondary endpoint, and it would be secondary prevention, because it would be the prevention of new cancers in men, women who had already had one cancer. And that is a different context. It will take a lot longer than the trial currently. We can't believe the trial's reported just after 2.5 to 3.5 years, but certainly in prevention settings. To look at a prevention effect, we're probably talking another 5 to 10 years before there are things that one could be confident about from those data. Signals will come sooner.
That is true. Now, I'm going to follow up on 2 points. One, to have an overall survival benefit at a follow-up of 3.5 years is kind of unprecedented in breast cancer. It rarely happens.
It's not happened since Herceptin.
Yes, and Herceptin was the last one that got a standing ovation at ASCO. So I'm sorry that we'll give it to you later. So tomorrow we'll give it to you. The second point I'd make is that you are completely right that any statistician would yell at me for what I'm about to say, but numerically there are already fewer second cancers in the olaparib group than in the placebo group in the study. That does not meet statistical significance, but certainly there aren't more. No. And that's going to be another issue that we discuss, that that we, there was a possibility that we hurt people. There was a possibility that we caused leukemia, that we made people sicker. But instead, there were careful patient-reported outcomes done. There's no difference in leukemia risk between the olaparib and the placebo. And, in the end of the day, these PARP inhibitors do, their DNA damage, they cause DNA damage. So we needed to be careful.
And Andrea, it was important that we would pre-specify. Again, it sounds like Susan and I have been taken into a back room by statisticians on many occasions and sort of whacked about the face because we are behaving.
Yes.
But it's very important that we had pre-specified some of these hypotheses around effects on second cancer risk, which we have. So I think we will be in a good position to ask it. The signals are all encouraging, and we're not harming people in a way that means that you could consider in a different setting, begin to consider, could this be a prevention strategy in lower risk people even than were in Olympia?
Right, and I, this, by the way, we're totally off script now, but this is good. So one of the, one of the real questions and one of the reasons it was great having Yaz here is that, we have to model some of this out too. So what would it look like to give prevention, and that, just as a reminder, tamoxifen is a drug that's approved by the FDA in prevention. It started for metastatic breast cancer. It went into what we call the adjuvant setting, early stage, high risk, then lower risk, and then a true prevention trial. And it got all the way through there. And so, but we give tamoxifen for 5 years. We're not going to give olaparib for 5 years to healthy people. So is it a week? Is it a month a year? Is it a month every 5 years? Because you can think about it as you just wipe out all those all those cells, right, that are about to become a problem. And how often do you have to reset? Is it like a colonoscopy that you just take out the polyps and you reset the situation and then you're 5 years later? So.
Yeah, I think with the, if I'm not wrong, Roger can correct me, but the carry or the heterozygote, if you want to model the heterozygote carriers, don't, in mice at least, don't tend to lose the other allele. So I think it's hard to, I don't know about modeling.
But I think doesn't Yaz's new model do that? Anyway,
well, I think he makes a knockout.
Yeah.
So different from carrier heterozygote.
Yeah, but it still would be, so you can tell, we're going to take this conversation into the back room later over drinks.
Yeah.
But you can see that we're intrigued by it and we're really starting to think about what would it take? What would those next steps be? Yeah.
Well, I think in those discussions, I think one of the very important things, especially when we're talking to patient representatives, is tamoxifen. You mentioned, these are drugs, even tamoxifen, drugs they're not without side effects. And what is considered acceptable in a prevention setting? What do women predominantly feel they want to take to prevent the occurrence of cancer and avoid prophylactic surgery? There are limits to that, what people are prepared to accept in terms of side effects. And PARP inhibitors are great, but they're not without toxicity. They cause fatigue, they cause low-grade nausea, and as you say, it's not the same as giving hormone therapy, which you might take in the long term. You're going to be pulsing it in order to remove the cells that have lost the wild-type allele, which is only going to happen intermittently. So we're at the first base in working out what the right thing to do. I totally agree. We need the model systems, mouse model systems, and those that look most like what really happens in women with founder mutations, for instance, not null alleles. And Yoss and Chris Lord and myself are trying to work on some of those models, others are too, that really mimic the founder mutations and might get reversions and the things that really happen.
Kara.
Yeah, I mean, I think that we can also get a lot of interesting information from the human patients, right? You already sort of mentioned sort of a pre-cancer atlas, and sort of Ronnie Dropkin has been pioneered in this work with an ovarian cancer to look at sort of these early lesions and see, do we see evidence in the breast of a woman with a BRCA1 or 2 mutation that there are different pockets, of the breast that might have loss of heterozygosity, seeing these early p53 mutations, because we don't really actually know the answer to that question, with sort of modern technologies and thinking about whole genome approaches and stuff. So I think that really could be the next space to know whether or not this is relevant in humans.
And it's why there's, collaborative efforts are needed, and, with Vassar and other Gray Foundation, recipients were working on this precancer atlas for BRCA carriers and making sure that we have the tissues so single cell sequencing can be done. And Kate's doing a lot of this work, and so it takes a village, because each independent sample is, you think they're common, but it actually is hard to get adequate human specimens to answer all the questions that we want to answer. So.
Can I just ask,
Sure.
or raise something, as this is sort of conversational? I think so many women are having to do that incredibly difficult thing of risk-reducing surgery, and there are so many, I think, scientific initiatives that could work with that tissue.
Right.
That is, we do so much more now that isn't just based on having frozen tissue or put it even in formalin, that you can work with live cells, take them into modeling, take them into mouse modeling. So I think initiatives that allow women to make sure that risk-reducing mastectomy material, much of which doesn't get looked at by, all of it doesn't need to be looked at by the pathologist, can contribute to research, are really important.
Yeah, so at Penn, we look at all the surgeons' schedules, and we reach out to all of our patients to try to, and so far our carriers about 60% agree. So, and we're trying to increase that. So yeah, exactly right. I mean, it takes every bit of this to work forward. So I'm going to change then. So PARP inhibitors. I mean, I've got a woman on a PARP inhibitor for 13 years. She was on the initial iceberg trial, Andrew. But most of the time, resistance develops. So let's talk about resistance. So Andre, you first. Talk about resistance.
Okay, so I guess in the clinic, as far as I know, the major mechanism of resistance is reversion mutations. So if you have a mutation in BRCA1, somehow or other gets respliced, put back together in a way that makes a functional protein. And I think dealing with that resistance is complicated. And I don't know that about, maybe you know more about therapeutic strategy there. That's a major mechanism. And the mechanisms that we discovered are more complicated, where you lose another DNA repair protein and you make the cell homologous recombination proficient again. And then I think there are several therapeutic strategies, because they're basically, bottom line is that there are many DNA repair pathways that they're redundant. So you can get homologous recombination proficiency through losing BRCA1 and 53BP1, as I discussed, but there's other pathways that you can then target to generate toxicity in those cells, the remaining repair pathways. But I think the reversion mutants are more complicated and more common.
Well, it's interesting, and I'll be curious to hear what others think. I don't know what the pie chart looks like, personally. As a clinician, I know that I can find reversion mutations because they're findable, because I can send a blood test and I can find reversions. But so the data that that I'm aware of, it's 20 to 25%. But I don't know what the rest of the 75% is. Is it reversions that I couldn't find? And what is it about all those other resistance mechanisms that many of which you've figured out? How do they play a role? And I think we have kind of case reports here and there of a PARP1 mutation or other things. And I think we really do have to figure out what the pie chart looks like, but it's hard. So Andrew, do you want to say something about the reversions? I know we're going to hear about this more tomorrow by one of your colleagues, so shout out there. Stick around.
Steve,
you can hear, stick around for tomorrow. But about those.
Sure. So I think one thing I think we really need to know much better, we know more about this in ovarian cancer than we do in breast cancer, is the frequency with which resistance is driven by reversion mutations, and where reversion mutations happen, are they coexisting with other resistance mechanisms? Because it's not a given that there will be one resistance mechanism in a tumor. Tumors can be clever and learn to be resistant in more than one way at once. And so we need to know that, because if we're focusing our treatment on one resistance mechanism, but two exist, we're not going to be so successful. So there's an awful lot that we can learn now from actually liquid biopsy, plasma analysis. There are ways that I think we can sample tissue at the point of resistance to understand these better. Reversion mutation, when we do understand how frequent it is, one of the approaches that, and Steve Petit has led on this in Chris and my lab at the ICR, is going to talk more about this tomorrow, is when you create a reversion mutation, I sometimes refer to them as fix-it mutations, where the BRCA gene fixes itself because of DNA repair, things that have gone wrong. It doesn't completely fix itself. It fixes itself in a way that creates a new bit of protein sequence, or leads to the visibility, the expression of a new bit of protein sequence. And these are the things that the immune system can recognize. These are what we call neopeptides, that the immune system could be engaged to fight against with the kinds of immunotherapy that we're now aware of more broadly. So taking that approach, using it as potential vaccine strategies or CAR-T strategies, possibly in combination with checkpoint inhibition, is an approach to targeting reversion mutations. And we've got some ideas about how that might be done.
And I do think it illustrates the fact that why a tumor becomes PARP inhibitor resistant or platinum resistant may lead to what you do next.
Yes.
So it's not a one size fits all. A tumor becomes PARP inhibitor resistant and therefore everybody gets the next thing. No, it may be that if it's PARP inhibitor resistant because of A, you do B, C, you do D. You know, it's going to stratify, and that's going to be really important for trials. And I think we just, again, don't have enough information. So Kara, I'm going to toss you the HRD question, which although I know Andrew loves to talk about the biker with the tattoo, well, I'm going to tell us, in ovarian cancer at least, we know that HRD, homologous recombination repair deficiency, can be measured beyond BRCA1 and 2, and this has been most clearly demonstrated in ovarian cancer. Can you tell us what is that? How does it get measured?
Right. I mean, to do this in front of Dr. Tutt, I don't know if can I do this? I don't know.
Yes,
Please, you do it.
So I mean, you know, it's such an interesting concept because it brings like all levels of how we could measure things. And so when you are what we call HR deficient, we think it's something that maybe we can just get a blood test and just kind of it'll tell us that. But there's actually, what we're actually doing is really looking at surrogate measures when we get a tumor. So we're looking at the response to not having that pathway in the pattern, the way the DNA looks. We can also do it by how the RNA looks. And so I think one of the big things that, Dr. Tutt and other people have been so important in describing is that the right way to do it, though, actually in the clinic, right? So is that surrogate measure the right way? Because it's not an actual functional assay. If you want to know whether or not your bike is working, you're going to go out and ride your bike. You're not going to find out, like, is there a pattern of, brake failure or something like that. You're actually going to want to see the functional aspect of it. So one of the things that I think is, could be really important is looking at the proteins that are necessary for homologous recombination and then saying, are those back on? Because that really is what matters clinically. And, I know that, and I don't know if Dr. Tutt wants to comment on this, there's assays that can do this. Yep. We do immunohistochemistry all the time. It's a type of assay that looks for proteins in tumors. So hopefully maybe we can do that.
So, thank you. I think the tests that we're talking about are essentially looking at the tumor and looking for a kind of diagnostic tattoo. Right. That says...
Oh, come on, you're going to do the biker and the tattoo, the priest.
I'll do the biker.
Do it.
I'm going to fail now.
Yes.
So a diagnostic tattoo. So you can look at the tumor and a type of tattoo is going to say there is homologous recombination deficiency. The problem is that, as we've learned from what Andre's been telling us, what a number of people in this room work on, that that can change over time. That DNA repair can be switched back on. But you can't get rid of the tattoos. So the Hell's Angel, who's covered himself in his Hell's Angels chapters, diagnostic tattoos, is going to have a big job to remove those. And yet, I've got a photograph from the internet of a guy who's clearly a Hell's Angel who became a Catholic priest. And I'm pretty sure that he's different in the way he lives his life from your regular Hell's Angel. And unless you're actually looking for the second level test, the presence of a dog collar, you can't tell those two men apart. And they are fundamentally different. So it's useful to do the first test. I want to know who's ever been a Hell's Angel. And then I want the second test to tell me who's still a Hell's Angel. And I don't think we have to have these. So this was the concept of the biker priest that Susan's referring to. Sometimes we want tests that are very simple. We just think, I just want one test. And in fact, I think what you need is to combine those two. You've got a filter, which is these really useful mutational signatures. And then you need something which just at your point about to use a therapy like a PARP inhibitor, you've got a functional test. RAD51 analysis, protein analysis can be done. There are other things that one could look for. Those are doable in the clinic. We need both.
So Andre, tell us, this is a very general question, but tell us why basic science is important. I think it's our hardest thing to convey when we're talking as a panel, because it's really easy when something works and we can trace it back and say, see, the people that were working on synthetic lethality in yeast, that's what got us to PARP inhibitors. But at the time, no one, maybe they didn't think that was so important.
Yeah.
So
I guess through basic research, you can understand, why loss of BRCA predisposes to cancer. You can understand why these tumors are sensitive to DNA damaging agents, and you can understand basic resistance mechanisms, what we're discussing.
But we don't always know. We go in a number of different directions before we get the straight path, right?
Certainly. 99% of experiments fail, and your predictions most of them are incorrect in biology. So just keep plugging away.
Yeah, that's why it takes persistence and patience, I guess, of that. One of the questions that we had submitted, which I think is really important. Oh, actually, one more for you. We talked about this all day, but just to bring it home, what are the potential new therapeutic targets based on biology for BRCA-associated cancers?
We're going to hear an important one tomorrow, apparently, about reversion mutations. Really looking forward to that. And then, again, a lot of work has been done on resistance mechanisms involving DNA repair pathways. To try to find vulnerabilities that still exist in the resistant tumors, and it's all about different redundant repair pathways. You get more and more deficiency in the tumor, and maybe the normal cell does not have that deficiency. So you're looking for vulnerabilities in the tumor.
So pol theta inhibitors are coming our way in terms of the clinic, things like that.
Yeah. So pol theta is another pathway that people have been working on, basic mechanism of repair when you don't have homologous recombination. You have this minor pathway that keeps the cell alive. So if you can target that pathway, you can kill the tumor cell. And since it's a minor pathway, it doesn't have much of an impact in the normal cell.
Yeah. So that's what we're always hoping for. Kill the tumor, leave the normal. That's always our goal. Andrew and Kara, this is, again, one of the submitted questions, and I'll be curious at your answer. How close are we to be able to screen for BRCA-related cancers using blood or other non-invasive testing? And by the way, my colleague, Mark Robson, who I think is up there, he'll be speaking about this directly, over there. He'll be speaking about this directly tomorrow as well. But any comments on our...
Okay.
Oh, I have many thoughts. I think that that there's been advances that seem to have pushed this right in front of us right now, and there's some tests that you can order to see if you might have cancer already. But I think that we just still need to be really, really careful. And my bias is that we're going to need multiple different levels of tests, that it's not going to be a one-size-fits-all approach. So the tests that are sort of, the sort of farthest along is taking a blood sample, isolating the plasma from it, and then looking at the DNA fragments that are in it. But you can look at those DNA fragments in a lot of different ways. You can look, are they broken up into pieces when they shouldn't be, or at more of a level than they should be? Can you look at what we call epigenetic changes or methylation signatures on the DNA to see if those are different? And what's pretty remarkable, that one of the companies that's working on this is that you can actually target those methylation signatures back to the tissue of origin. So that could be pretty cool. But we've used, protein-based assays for early detection for a very long time. We use imaging as well. So likely it's going to have to be a combination of all of those things together, in my opinion. But I think it's going to come really quickly because I think there's a lot of push to do so. We just have to be very, very thoughtful in how we analyze that data.
Yeah, I want to differentiate, these tests and how we use them, because if we have an individual with, say, metastatic breast cancer, we send these tests not infrequently because we're looking for potential therapeutic targets, like something called PI3 kinase mutations, where we have a drug called alpelisib, which we can give if we find that. So that's an example of a very established way to use the test. There's nothing controversial about that. When you take it the next step in, and Andrew, I'd love to hear more of your thoughts on this, if you take an early stage breast cancer patient, but who's at high risk for their cancer to come back, is there a role for looking then at whether you can detect anything in the blood? And I don't know if you want to comment on that.
It's not my own expertise,
Yes.
but I have a colleague in our centre, the Breast Cancer Now Centre at the ICR, Nick Turner, who's done a lot of work in this area. And as you say, the idea that taking a personalised approach to tracking in the blood, where you might take somebody's cancer, look for the particular features of it, it's then been removed surgically and they've had all of their radiotherapy treatment, their chemotherapy, but some people at least are at risk of recurrence and you really need to add lots more therapies, and some people may have needed no more therapy, and we just do it at the moment based on risk, population risk. I think what you're referring to is the idea you'd test in the blood and say, we can see that there's still something there at very low level based on a personalised test of the tumour, and then you focus all your efforts on the people who've got that sign and can you prevent this coming back. The trials are beginning to say that yes, you can predict those who are going to get recurrence very early. What they don't yet say is whether we can do something about it in an effective way. So this is, in my opinion, and I think Nick would support this, not ready for prime time, but this is incredibly promising as a way of asking the question in trials, could we actually prevent people developing that inevitable recurrence by bringing in therapies earlier than waiting for them to get that recurrence? And we're I think, really on the threshold of those trials being done and giving
they're
answers.
ongoing.
They're
and,
ongoing.
And we'll see. But I feel pretty strongly that they shouldn't be sent off trial because they're messy. It's a mess.
It's a trial, yeah.
And likewise, for early detection of disease, we do a really good job detecting breast cancer, and these tests don't really add to that. And so the question is how much these tests are going to add to things like pancreatic cancer and ovarian cancer screening. And so far, all we have is what we call case-controlled data. We don't have prospective data, and the prospective data that we have is, I think we're all enthusiastic about these tests, but I think that there's issues with them, and the one test that reported out of the people with a positive test, more than half of them, they couldn't find the cancer. So just put yourself in that position for a minute and think about what that would be like. So we have to have better tests, but I think we'll get there.
And I think one of the things I think people are increasingly understanding is sometimes you have a change in DNA in the blood that you might think, oh, that's definitely a cancer sign.
Right.
And in fact, it isn't. It turns out that some of us have got, I was talking to Bob about this earlier, some things you absolutely think are only going to be present in an aggressive cancer, that it turns out we all have a little bit of that mutational change in our makeup anyway, somewhere in our bone marrow, and it's never going to do anything harmful at all.
Yeah.
And if you're then seeking to have, where is that cancer, which bits of my body am I going to have removed, that wasn't a useful test for you.
Yeah, and don't worry, Kara, I'm going to set you up for it because I choose that. So this is what Andrew is referring to, and just to have a new vocabulary word for you all is CHIP, or conohematopoiesis of indeterminate potential. And it turns out we're all a little bit mosaic.
Yeah,
so
exactly.
Tell us what that means, Kara.
Yeah, I mean, I think this has gotten to be, so because I study patients and I see patients with p53 mutations, I also see patients who have p53 mutations that they weren't born with. And so this is a process called clonal hematopoiesis. So, I actually remember, and I'm going to shout out to Dr. Schuchter, who's our division chief here, because I think you might have taught me this very early on in my fellowship about the inverse relationship between age and how much bone marrow cells you have, right? So you take 100 minus your age, that's how many bone marrow cells you have. So your bone marrow is constantly sort of going down as we get older. And as it decreases, you have the possibility that a mutation could cause a selective advantage. And then you're gonna see that in your blood. But just because it's causing a selective advantage and that cell happens to be living, doesn't mean it's actually doing anything bad for you. And so we're, not really sure with all this genetic testing we're doing, we're starting to see these low-level mutations. And even a gene like P53, where, when we tell the leukemia doctors, right, when we tell them we have a P53 chip, they're all like, oh my God, that's terrible. But we don't actually know that that's terrible yet. So I think that, that's really something that's going to be important to figure out.
Yeah, and especially as these tests get better and better, and we detect lower and lower and lower levels of this, it gets a little tricky.
It gets a little tricky.
So I'm gonna ask you guys a big question, and then we're gonna take questions from the audience, of which we have of course a bunch here and there. So, but first, Andre, tell me, what do you think the next big step is for BRCA research, for you? What's going to be the thing that pushes it the next step forward?
Well, I think I am, as I mentioned in the seminar, manipulating homologous recombination, changing homologous recombination proficient cells to deficient cells, increasing vulnerability, converting them to microsatellite unstable. That's the kind of thing that we're starting to work on, as in my talk, which might be relevant to these reversion mutations as well.
Just this idea that cancer cells aren't set on their path. Maybe we can push them in a direction we want them to go in to make them more susceptible to treatment.
Yeah, exactly.
Okay, Kara, how about you?
Well, it's interesting. I mean, I trained as a basic scientist, and so I was trying to think of what's the big basic science question I have. Trying to figure out how we get this genetic testing to be more widespread available has been what's been keeping me up at night. And now that I've done a lot of work in the VA and sort of seeing the way that lots of things are in this country, it's really hard to get genetic testing to everyone, and this more than anyone. And I think that we need to push this forward. We need to have more education, we need to have more accessibility, and then we need to be able to have, more to be able to do about that. And we have the workforce, we just don't always have the education for patients and for, particularly men, to understand how important it is.
Yeah, not just men, I want to give and another specific question was placed about, diversity and sort of equity. And we know that this is not something that we do particularly well in the United States. We know that fewer women and men of color undergo genetic testing, even for the same level of familial risk. And so it's a very keen thing that we're aware of, that we're working on the Vassar Center with a lot of support. We have a scholarship, we have an underrepresented minority program for the summer. With the Cancer Center, there's a major initiative for clinical trials to make sure that within our catchment area that that we have appropriate representation, so that we can answer the questions for everybody.
And one thing that I'll talk about this a little bit tomorrow, but some new data that I've been working on with one of our fellows, suggests that in equal access, setting like the VA, we don't see the same disparities. So that's kind of cool, right? So, I think that that's going to be really important.
Well, we also know that when Medicaid was expanded in the United States, the states that expanded Medicaid, all sorts of things, all sorts of disparities shrunk. Time to initiation of treatment, therapy. So we have an insurance problem in this country, and until we fundamentally fix that problem, it's going to be an issue. Having said that, we also know that patients aren't always even if they bring it up, their doctors don't always recognize them as good candidates for genetic testing, and so they aren't referred. So this is a multifactorial problem. We need to educate providers, we need to educate physicians, and we need to make sure that our amazing genetic counselors, do the jobs that they're so good at. Okay, Andrew, we're up to you.
So I guess, and forgive me of just focusing on where you capitalize on this result in early disease, early breast cancer. So I try and think about that in two ways. One is where could we, having learnt that, de-escalate treatment and make it less onerous for often very young women? Can we actually give less chemotherapy by bringing this PARP inhibitor tablet-based approach in? Can we take bits of our very toxic chemo out and put PARP inhibition in their place? For some people, maybe no chemotherapy at all. Maybe we could rely on a PARP inhibitor, but one size fits all is going to lead to a disaster. So that's where biology needs to come back in again. Where do we really feel we can de-escalate strongly, and where do we need to understand resistance quickly? The other side of that is the resistance. People now will be getting PARP inhibition early in their disease, and that means that we need to be on it in advance to, if they do get a recurrence, where are the right approaches? So it's a kind of twin track to the research. And the third element is, I know this is less relevant to the genetic context of BRCA, but as Serena Zainal said in her lovely talk earlier, actually this targeting homologous recombination deficiency applies outside heritable cancer. There's a lot that's driven by what we call epigenetic changes, and it would be a quick win to move into that, as long as we're careful to understand that can change quickly. That what we call epigenetic BRCA-ness could be much more plastic. It might change from being the Hell's Angel into the biker priest more easily. So we need to do that carefully. But if we do it carefully, we could extend the benefit to a really quite large chunk of cancer.
Just two follow-up points on that. One, the de-escalation point, because de-escalation, is always kind of a scary term. That we're taking away therapy that we knew worked, and we like to. I think it's really important that as breast cancer doctors, we've seen the field change so significantly. Certainly, when I started in medical oncology, if you had an estrogen receptor positive tumor that was over 1 centimeter in the United States, everyone was offered chemotherapy. And nowadays, if you have that same situation with node-negative cancer, estrogen receptor positive over 1 centimeter, 75% of women do not get chemotherapy. And that's on the basis of large trials that were done showing that we have this other test. We use Oncotype, you use Mammoprint, that shows which tumors don't need chemotherapy. So we just need to do, it's the same approach. We're doing that with HER2/neu. Can we do it with PARP inhibitors? And I think it's just going to be really interesting to sort of see where we go, because we're not medical oncologists and we give chemotherapy, but we'd rather not. So if we have something else to do, it's all right with us. We don't need to do that. The other comment, just to follow up on this HRD and then BRCA1 and 2 thing, is messy, because on the one hand, not all tumors that have BRCA mutations have HRD. And the specific example Kara gave, which is you might happen to have lung cancer, potentially because of environmental factors and heavy smoking, and just happen to have a BRCA1 mutation. They're true and unrelated. So your tumor is not necessarily going to respond to that. And there's been data that show that a lot of these, what I like to call non-canonical tumors, are atypical atypical tumors that are not generally associated with BRCA1 or 2, most of them probably don't respond very well to BRCA-directed therapeutics. But we need a tumor marker because some of them might, and we don't want to miss them. And on the other hand, as Andrew mentioned, again, ovarian cancer is a different beast, but in breast cancer, and Andrew's been working on this for a while, what are the appropriate assays to determine which patients, will go forward? And I think, what Serena's work is extremely interesting, but as Kate mentioned, right now it requires fresh tissue, which is challenging. So hopefully, you'll solve that problem.
So with Serena's, I just want to see if I understood the Serena's work. My understanding was that there were, in breast cancer, a lot more than expected are HRD.
Yes.
She said, I was surprised by the frequency. Did she say 1 out of 5 or something?
So in breast cancer overall, about 20, I think she said 22%.
That's much higher than...
But in
I
triple
thought.
negative breast cancer, it's 60%.
Yeah.
That's also much higher than
But
I
the
thought.
majority of the cause of that is not genetic.
Yeah.
It's BRCA1 or RAD51C methylation. And that's why I think it's really important we think about extending into that. But that's where it could be that changes quite quickly. They lose their promoter methylation. And that's not hard to work out. It's just we need to be careful and use the test, the HR, the signatures. And I think what these techniques of whole genome sequencing, which are becoming so much cheaper to do, it may be challenging in formalin-fixed material, but we have RNA later. We have things that are very easy. You can still stick it in a tube and leave it for a month. It's just we need to stick it in a different tube than that which our process currently tells us. So go figure, change.
But one of the real worries, and I swear we're going to do questions next, but we're having too much fun, is that in triple-negative breast cancers, what we've managed to do over the last few years is just add and add and add and add and add more therapy.
Yes.
And right now, if you're diagnosed with a triple-negative breast cancer, in the United States, you get AC, carbotaxol, and pembrolizumab. And it's a lot of therapy, and so. Just as importantly, it's identifying the people that we may be able to do less. So, all right. So that was fun. I'm going to now start with questions. Again, I'll keep close track on the times. I promise we won't go over. I'm going to take some of these. There were a few different questions about pre-implantation genetic testing. Just as a reminder to everyone what that is. If an individual, either a man or a woman, has a BRCA mutation, and they wish to have a pregnancy and have a child that does not have the BRCA1 mutation, they can go through in vitro fertilization, screen the embryos, and only reimplant the embryos without the gene mutations. And in general, if people are interested in having a child without a BRCA mutation, that is the way that we know how to do it right now. Some people ask about CRISPR. We are not CRISPRing embryos at this point. But embryo selection is sort of the thing that is offered to individuals. And so it is also a reason we often get the question, and it's in here too, when do people get tested? In the United States, if there's a known mutation in the family, we test the women at age 25 because we start breast MRIs at 25. In the UK, it's a little different for BRCA2, it's 30, but in the US it's 25, so that's when we say the women should be tested. For the men, and Kara's gonna figure all this out for us, our recommendations for men are not as evidence-based as we would like, and so usually we're not starting stuff till 35 or 40, unless that man would like to use that information for reproductive purposes. And so if they're about to start their family, then a potential reason to get tested is for that reason, to start to see if they wish to do a PGT. So that's that one. Kara, I'm going to toss you pancreatic cancer screening. How do you do it? Should you do it? Who should do it? And I know Bryson's here somewhere, but you give a start.
I'm not sure if he was going to be able to get back. I told him I was going to be giving a shout out. So Bryson Katona works with us, quite significantly in this space, and so he's done a lot of the work in pancreatic screening. And so, as folks know, and I don't know what you guys do in England and the U.S., we do either endoscopic ultrasound or an MRI, and I think a lot of that depends on access and the availability of what's called an advanced endoscopist. that is necessary to do this endoscopic ultrasound, which is just like as it may sound, a camera that goes down the throat. There's a little ultrasound probe that looks for the pancreas through the stomach, and they can look to see, is there something that might be concerning? And the beauty of that approach is that you can actually stick a needle in that something if you see it. An MRI, of course, you wouldn't be able to do that same thing at the same time. So, but these are invasive procedures. I think there's still a question of how frequent, how long, what age do you start? And so one of the kind of cool things, like I mentioned before, is can we do more than one thing at the same time? And so there's really neat data suggesting that a pre-diabetic state could be an early precursor or an early marker of pancreatic cancer, particularly if you see this elevation in this marker called A1C, which suggests is a marker of how high your sugars are in a patient who's not gaining weight or in fact even losing weight, that could additionally help screening for pancreatic cancer. And Basser's obviously been very involved in those studies.
So the current NCCN guidelines, so in the United States, the guidelines are that if you have a BRCA1 or 2 mutation or some other genes, but focusing for BRCA1 and 2 for the moment, and you have either a first or second degree relative with pancreatic cancer, pancreatic cancer screening starts at 50 or 5 to 10 years before the earliest pancreatic cancer in the family. The role of pancreatic cancer screening in individuals that don't have a family history is controversial. We do offer it on study at Penn, but it is a controversial area. And I think in Europe, that again, things are evolving in Europe too. I don't know, Andrew, if you wanted to say anything more.
It's not a particular area of expertise, but I don't think we are quite as aggressive in our sort of screening strategies for pancreatic cancer as is here. I'm not aware that we're certainly doing kind of interventional screening. It's more imaging based.
And we just don't have the answers, and it has to be yearly, and so we want to start at the right time, because we do know that people get screening fatigue pretty much of anything, and so if you don't start at the right time, you might end before you were ever at risk. So we're really trying to sort those things out. Okay, and someone did ask about someone who is 39, and we wouldn't start we would not start that young unless there was a significant family history. So I want to just say a few words, and maybe some other folks could opine too on the prevention question, because cancer interception, prevention, these ideas, cancer interception is the idea that we intervene. So you have one bad copy of BRCA1, and just when that second bad copy hits, before you even really have a cancer, maybe that's where you can act, right? That you can intercept the cancer at that time. And so, we are starting to have some really tangible ways to think about cancer interception or prevention. So one large international trial that is now enrolling, and we're about almost have it open at Penn, but it just opened in the United States, is called BRCA-P. It's looking at a drug called denosumab. Denosumab is a rank ligand inhibitor, and this is a medication that is used, that is FDA approved for treatment of osteoporosis. It's been studied previously in breast cancer in terms of protecting bones and potentially decreasing breast cancer recurrence. And there's been incredible work done by Jeff Lindeman, who's been here previously from Australia, and Christian Singer, who's in Austria, looking at the fact that rank ligand is really important in the development of BRCA1-related triple-negative breast cancers. So this is a large, more than 2,000 women, randomized trial, international, of denosumab versus placebo injection every 6 months in BRCA1 mutation carriers to see if it decreases breast cancer risk. But that's what it takes, by the way. It takes those kinds of studies. And so nationally in the US, it's led by Judy Garber. I'm on the executive committee. It's an important study. I know it's open in the UK. And we'll see. We don't know if it'll work, but we have to try.
Just to comment, at the moment it's potentially open in one site in the UK because of the challenges of doing these kind of studies and how they're funded, because these are academically funded studies. And yet they're so important, and they will learn. I mean, the biology behind that study is very, very strong. Jeff Lindeman, Jane Visveda, beautiful biology around this. But even though that trial is about that specific intervention for that biology, what you learn from it tells you so much about the approach of prevention trials
Yes.
for this particular demographic of women. And so we have to do this in order to learn what people want and what they'll tolerate for the next thing, whether it's PARP inhibitors or whatever it is. But at the moment, it is all academically
I'm
funded.
so glad you brought that up. So Andrew is also alluding to the fact that the Olympia trial, which he had mentioned earlier, this large adjuvant trial, was a very complex study of pharmaceutical company, the big international group in Europe, the NRG in the United States in terms of cooperative group, and it was complicated, to move things. It's still complicated, but but it takes a lot of money to do these trials, and it takes a lot of international cooperation. So this denosumab trial, for instance, Amgen Europe is funding it like in mainland Europe.
Yeah.
And then in the United States, it's funded in part by a grant Judy Garber has from the DoD, and also the Gray Foundation has provided costs for patient-related costs, because we basically have been cobbling together the funds, to do it. I don't know how you're funding it in the UK, but
It's charitable funding.
Yes.
The pharma company provide drug and things,
Yes.
some of the costs, which is in itself a big investment. But I guess the point is that to take these things forward on behalf of the community, it takes big sort of grant funding to do it.
Yep.
And then when that's there, it really needs cooperation and engagement for our patients to find this, to find the
Yes.
opportunity.
Yep.
And I think we're now in a much better position to say, this community know there's something different and special, and there's a trials network, and don't just treat me as someone who's got breast cancer or ovarian cancer. Overall, I've got something particular, and helping people find the trials because they're there. We're getting there.
in our beginning days, Andrew and Mark and I and others wrote some nice fiery editorials about not abandoning the BRCA population, because actually the PARP inhibitor programs were almost stopped. And part of the reason was it was felt like, there weren't enough patients, and we're like, and
It's not a big enough market.
It all worked out, but I mean, I think that you're right. We've made some progress. So we've got denosumab for prevention. You've heard a little bit about how olaparib, may or may not, got a lot of work to do, but PARP inhibitors in general, I should say. And then at Penn, we do have a vaccine study that we've been working on. We've been giving vaccination against telomerase, WT1, and PSMA with electroporation, plus or minus IL-12, with a first cohort of individuals, BRCA1 and 2 mutation carriers with cancer. And we have, the go-ahead by the FDA to vaccinate healthy carriers, which, we hope to start soon. We are through our halfway through our first cohort, and then we'll start our healthy cohort. So we have some really tangible things that we're doing. Somebody asked about supplements. Supplements are complicated in prevention because some of the prior studies, like in prostate cancer and lung cancer prevention using supplements, were not only negative trials, they didn't only not prevent the cancer we wanted, but there were actually harms associated with them. So supplements are really messy, in part because we don't always know what people are taking. But in those studies, those were even controlled supplements. So, I'll be honest, except for vitamin D, living in Philadelphia in the winter, and making sure you have enough calcium in your diet, there haven't ever really been good studies that supplements are particularly helpful, and that actually just eating good healthy diet, minimizing alcohol, and regular exercise, that's the kind of trifecta of what we've got environmentally. I don't know if anyone wants to add anything to that. Again, I'm just going through, we got 8 minutes and more questions, so we're just gonna keep going. How about a question about variants of unknown significance? So an individual says, I have a BRCA variant, I had breast and uterine cancer, but it's not associated with increased cancer risk. Should my brother get testing? So Kara, what is a variant of unknown significance, and how do we handle that clinically?
Yeah, so we all, as we've probably, you may have heard, we're all born with lots and lots of variants. That's what makes us all look different. We had a great talk earlier by Kara Bernstein talking about efforts in certain genes and trying to figure out what do all those variants mean, because it's really, really, really difficult to use a computer even to model what a variant does or does not mean. So when we have certain types of variants that you can tell just disrupt the whole function of the protein, that's easier to tell. But some of these other variants that maybe tweak it, I always like to use the example of a book. If you remove a chapter of a book, you know the book isn't going to be worthless anymore. But if you change an A to a T in one word, the book's probably fine. But maybe that's a really important word that when you change that letter, it actually does disrupt the function. So right now, I think there's a lot more variants of uncertain significance in all genes than there are variants of known significance. Now, BRCA1 and 2 is a great place where that's actually not the case, and we do know a lot about the variants in those genes, but there's still obviously a lot of work there. But I think the most important thing, and the thing we talk about over and over and over again, is not to make treatment decisions or any kind of decisions based on a variant of uncertain significance, and that does not have the same implications as an actual mutation. One thing I'd like to comment on with that is that as we do all this more sequencing, and I love hearing all the whole genome stuff, we and others have shown the number of variants of uncertain significance you get is going to go way up. And we're certainly seeing that as we do more and more genetic testing. So it's not going to go away.
Yeah, and if you have a variant of unknown significance and you haven't checked in with your ordering provider recently, just do it, because a lot of times, 95% of the time, they're reclassified downward as unimportant. Whoever ordered your test should be reaching out to you if it's reclassified. But, it's a two-way street. Sometimes you move, we can't find you. So, we always ask people to kind of check within us periodically. Next question was about a partial mastectomy so you can breastfeed. And here I'm just going to be honest, it's a little bit all or nothing. If you only remove part of the breast, we don't have any really good evidence that would really reduce your risk. I mean, it might sound like we would know that that's true, but we don't. You still need MRIs. So in general, it doesn't really help very much because you're still going to get intensive screening. Somebody asked about nipple-sparing mastectomy. Now that's different because we just leave a very small amount of breast tissue behind. And my terrible analogy is that when you get a mastectomy, you never take every last bit of breast tissue anyway, because it's not like opening up and taking out a baseball. It's more like a bowl of Jell-O dumped on a shag carpet, and then you're like scraping the Jell-O off the carpet. So the skin layer, if you scrape it too much, you hurt the skin, and the skin dies, and that's not what you want. So we always leave a very small, the surgeons, leave a small amount of breast tissue behind. And nipple-sparing mastectomy, the amount of tissue that's left behind doesn't significantly add to that. So the studies so far have not suggested an increased risk of breast cancer or post-mastectomy breast cancer recurrences after nipple-sparing mastectomy. It is to say that mastectomy decreases the risk of breast cancer by more than 90%. It's not 100%. There's approximately a 1 to 2% risk of developing a breast cancer after you've had a mastectomy. But I like to remind people that that's much lower than your neighbor, if you will. And nipple-sparing mastectomy, women in general are much happier with their cosmetic results. So it's something to talk to your surgeons about, but it's not contraindicated, from my standpoint. There's a lot of questions, a few different questions about, I've already had my mastectomy and I've had my ovaries removed, what else should I do? And this is the question we get a lot. So I think that we do have these conversations about pancreatic cancer screening, but again, it's complicated. It's not a one-size-fits-all approach, but I think that is going to change. The next 5 years are going to sort of bring us a lot of changes in that. Someone asked us about colon cancer and risk of beer, and that actually has been somewhat controversial. And Stephen, you're here, and you can certainly chime in. I think that the data, Dr. Nayrod is here, and there have been some initial data. There's some small increase for colon cancer in BRCA1 carriers under age 50, but in general, the effect size is small. Now in the United States, we do a colonoscopy at 45 anyway, so it doesn't necessarily, change medical management as long as there is no family history. Stephen, go for it.
Paper Kara spoke about came out this week with Anthony. They had a lot of data on COVID. It didn't seem to show anything for BRCA1 and non-significant for BRCA2.
No?
Yeah, exactly.
That's right.
Yeah, so I would take it off the table.
Awesome. I love that. We just took it off the table.
Regular screening.
Yeah.
But then the flip side is that,
Yeah, exactly. So we're at 3 minutes away, and so I want to first, I didn't get to all your questions, as we always do after this. We do, for the Bassler Center, I answer a bunch of these questions by video, and we post them on the website. So if your question was not answered, I promise I will get to it. I hope you've enjoyed the panel. I want to thank our panelists for excellent work, and I'm going to introduce Dr. John Epstein, who's going to take us home. So.
What a fantastic day. I'm John Epstein. I'm the Chief Scientific Officer here at Penn Medicine. And how about another round of applause for all our speakers today? Also, my congratulations to Dr. Shah for winning your prize for the Pearl and Phil Bassler Innovation Research Award. Pretty cool to win an award from Philadelphia Phil and Pearl Bassler. It doesn't get a lot better than that. Sherry and Len, thank you so much for supporting the Global Prize and for the terrific seminar and winning the Global Prize, Dr. Nussenzweig, and for all your support of the Bassler Center. We heard a lot at in the introductory comments about Sudoku, about giving back. We've had 10 years of advances through the Bassler Center, thanks to the Grays and their friends and their interest in giving back. That word in Hebrew has another meaning. It's also the meaning of righteousness, and I think that's appropriate in the case of the Grays. In fact, I think there must be a Yiddish phrase that's something like a lot of tzedakah, and that phrase might be reserved for the Grays. We're incredibly appreciative. Here at Penn, I'm biased, but I think we're actually the best academic medical center in the world right now in translating fundamental discoveries into real cures. And the Bassler Center epitomizes that, taking importantly, the broad view that if you don't have some new discovery, you got nothing to translate. And if you think about why we start at the very beginning with homologous recombination and how BRCA works, it's hard to fix things if you don't know how they work. And the point of studying that is to figure out what an easier way to fix the problem will be. But you've also got to take it all the way through the difficult trials and prove that it really works in people. And the Bassler Center is doing that, case by case, example by example, chance by chance, to see if we can find new cures. And it's slow, but it's being successful, and we've made a difference. And Susan, you deserve enormous congratulations on all you've accomplished in this center. I'll say just one more thing, and that's that the eminence of the Bassler Center, I think, is now appreciated worldwide. We heard that in some of the questions earlier today. The Global Prize, the publications, the FDA approvals, this has resonated, throughout the field, and people in breast cancer, ovarian cancer, genetics of cancer recognize what's going on here. But the ripples are greater, and what I've noticed recently is that when I'm interviewing candidates who want to join the faculty at Penn, it's not just the people working in BRCA or homologous recombination who ask about the Bassler Center. Just the other day, a candidate who's working on computer analysis of protein folding and had never worked on BRCA before asked, "What could I get involved in this when I come here to Penn?" And to me, that's just an example of both the impact and the potential. Because if we can bring new and fresh ideas and fresh technologies to attack these questions, I think that's how we'll be able to take things even to the next level and make fundamental changes. So with that, I think I'll invite everybody to join us at the 10th year celebratory event that will happen just outside here. We'll have some additional comments and some refreshments, and thanks everybody for participating.